The question number one, which feature is visible with the light microscope using natural light source? So natural light is coming in a visible spectrum, contain 400 nanometer of wavelength. Half of this 400 is the 200. So any object which is less than 200 is not visible by the light microscope. Why? Because normal visible light cannot reflect back after colliding to this object, which is less than 200 nanometer. So that's why the option A, the DNA molecule, and C, the phospholipids, and D, the ribosomes, their size are less than 200 nanometer. So we cannot see these objects by the light microscope, while the paramecium cell of the diameter of 200 micrometer, which is more than 200 nanometer. Why? Because 1 micrometer is equal to 1000 nanometer. So that's why we can see the paramecium cell by the light microscope. So B is our correct answer. The question number two, the electron micrograph shows the structure found in the cytoplasm of animal cell. So we can see here that clearly the triplet tubules are there. So there are 9 triplet tubules which are arranged in a specific manner, and that's making centriole. So this centriole is making basal body of flagellum and cilia. So which molecule, which biological molecule are found in this structure? So we can see this centriole consists of 9 triplet tubule, and these are made of tubulin protein. So this tubulin protein, there are 9 triplet tubules with the tubulin protein. They're making microtubules, and they're joined together, and they're making this structure centriole. So nucleic acid is not present in that, and phospholipids are not present. So that's why the 2 is our correct option here. So that's coming in D. D is our correct answer. The question number 3, which cell structure contain nucleic acid? So this nucleic acid mean DNA or RNA. So cytoplasm, this contain messenger RNA, ribosomal RNA, and transfer RNA for protein synthesis. What about mitochondria? This contain circular DNA and messenger RNA. They are produced from circular DNA. What about lysosome? It contains hydrolytic enzyme for the intracellular digestion or extracellular digestion. So that's why there's no nucleic acid present in lysosomes. So C is our correct answer here. The question number 4, the mitochondria are thought to have evolved from the prokaryotic cell that are ingested by the ancestral cell. So according to endosymbiotic theory, so ancestral cell, they ingested the bacteria, and some bacteria, they were performing photosynthesis, and later they became chloroplast, and some bacteria, they were producing energy. So later they became mitochondria. So which feature have prokaryote lost during their evolution into the mitochondria? So what feature they were present in the ancestor bacteria, and right now they're not present in mitochondria. So we're going to see that, for example, the cell wall is present in the form of peptidoglycan. So that's present in the current bacteria, but this cell wall is not present in mitochondria. So that's why this character has been lost. What about the circular chromosome? So there is a circular DNA. This is present in the mitochondria. So this is a conserved structure. It is not lost. So endoplasmic reticulum, it is not present in the prokaryotic cell or in bacteria because no membrane-bounded organelles are present in prokaryotic cell. So that's why it is not present in ancestor, it is not present in mitochondria. While the ribosome, so it is a structure, conserved, evolutionary conservative structure that contains 70 Swedberg ribosome. This is present in mitochondria and it's present in its ancestor. So A is our correct answer here. The question number 5, which polymer are present in all viruses, all prokaryotes, and all eukaryotes? So first we have a polynucleotide, and this polynucleotide is a DNA or RNA. So that's present in all parts. For example, viruses, they contain in the form of genetic material. So a central core is made up of either RNA or either DNA. And the prokaryotic cell, they contain circular DNA in the form of plasmid, or they contain nucleoid region. What about in a eukaryotic cell? They contain DNA and RNA. They are present DNA. They contain DNA in the form of chromosome in their nucleus. So that's why this is present in all. What about the protein in the form of polypeptide that's present in all? So virus contain outer coat is made up of the protein, and bacteria contain peptidoglycan cell wall. What about in a eukaryotic cell? This protein is present in embedded form in the cell membrane in the form of enzyme and so many other parts. So that's why this is present in all. What about polysaccharide? This is present in prokaryotes and eukaryotic cell, but it is not present in virus. Like, for example, cellulose, starch, glycogen, lignin. So these are carbohydrate polysaccharide, and they're not present in virus. So our correct option is 1 and 2 are correct. So B is our correct answer here. The question number 6, how many of the listed structure typically contain genetic material that has telomeres? So a telomere is what? It is a repeated sequence of nucleotide. This is present at the end of the DNA or at the end of the chromatid. So this repeated sequence of guanine and cytosine, it prevents the gene loss during the replication of the DNA. So that's why this is an important role of the telomere. So the bacteria, the bacterial cell cannot contain telomere. Why? Because the bacterial cell contains a circular DNA. What about there's no N in the circle? So that's why there's no telomere in bacterial cell. And in the same way, the chloroplast and mitochondria both contain circular DNA, so there is no telomere is present. What about in nucleus? We have a linear DNA, which is making chromatid. These chromatid link together and they're making chromosomes. And these chromosomal end, they contain telomeres. So you can see end of the chromatid. So there is a repeated sequence of nucleotide, mean guanine and cytosine is repeating again and again. So this prevents a gene loss during the replication of the DNA. So if there's any part of a gene is lost, so that's why the cell cannot enter in a division phase. So that's why any cell who lost the telomere, that will become a specialized cell and cannot divide. So here, correct option is nucleus. So A is our correct option. The step number 1, 2, 3, and 4 are used to test of non-reducing sugar. So if you want to test non-reducing sugar, we take a sample of 5 centimeter cube of a solution in a test tube. That's a non-reducing sugar. Then we add a few drops of acid in that to hydrolyze the glycosidic bond. Why? Because polysaccharides are non-reducing sugar. But if you break down into the monosaccharide, so that will become reducing. So for hydrolysis or breaking down of glycosidic bond, we need heat. So we need to heat it after 2 steps. Okay, so neutralize this with the alkali. And the next step is we need to neutralize the acid. Why? Because Benedict solution cannot work in the presence of acid. So we need to neutralize it completely by using sodium hydroxide alkali. And step number 4, add the 6 centimeter cube of a Benedict solution then, and then we have to heat it to up to 90 degrees Celsius, or we need to boil it to get a positive reserve with Benedict solution. Now, when is the solution need to be heated or boiled? So we need to be heated after the step number 2 for hydrolysis and after the step number 4 to get the positive result with Benedict solution. So step number, that's option number 1, A, is wrong. Why? Because he's saying like we need to heat it between 1 and 2. So before adding to the acid, heating is useless. No need to heat that. What about we need to heat it between 2 and 3? So for hydrolysis and after 4, so B is our correct option. What about the option C is wrong between, we need to heat between step number 2 and 3 only. So if you will not heat after the step number 4, so we cannot reduce properly the copper +2 oxidation state. So when the copper is coming from the Benedict solution, which consists of copper sulfate. So for properly reducing the copper to get the positive result of Benedict solution, we need to heat it until the 90 degrees Celsius. So that's why it is the wrong option. And after 4 only we're supposed to heating, and the D is the wrong option. Why? Because if you will not heat it properly after adding the hydrochloric acid, so there will be no complete hydrolysis and the result will be not perfect. So that's why the B is our correct answer here. The question number 8, which feature contribute to the function of cellulose molecule? So that's a knowledge-based question. You should know that the characteristic of cellulose. Cellulose is a long, unbranched, linear chain that formed by beta glucose by the linking of 1,4 glycosidic bond. And every molecule of the beta glucose have 180-degree alterations, mean up and down. So these linear chain molecules, they are attached by the hydrogen bond, and it is insoluble in water. So this type of general information, you should know that for solving this question. So option number 1, we have a long chain of glucose molecule coil into helix. So that's a coil into helix is a wrong word. It is linear molecule. It's not coiling. So number 2, the many hydrogen bond form between adjacent chain. So this is correct. So these adjacent chain are connected by the hydrogen bond and it is insoluble in the water. So the statement number 2 and 3 are correct. So C is our correct option here. The question number 9. What correctly describes triglyceride? So that's a normal lipid, triglyceride, and this triglyceride is non-polar molecule that are soluble in ethanol. So this is correct statement. It's a non-polar molecule and it is insoluble in the water, but it is soluble in ethanol. So B is a wrong option, a non-polar molecule that are soluble in the water. It is not soluble in water. The C is the wrong option, and they're saying like polar molecule. So it is not polar molecule. This is non-polar molecule. So that's why it is, and it's soluble in ethanol. That's fine, but it's a non-polar molecule. So D is also incorrect here. So we're going to proceed to the question number 10. The cocoa butter contain 3 different triglycerides. These triglycerides are made from fatty acids. When we have oleic acid, palmitic acid, and stearic acid. So these 3 triglycerides found in a cocoa butter they're present in a 3 sequence, mean a POS, SOS, and POP. So we're going to see that what is the difference in that structure. So you can see this structure. For example, we have 3 fatty acids. They are connecting with the glycerol by the ester linkage. So they're making 3 different ester linkage, number 1. And in this way, first we have the palmitic acid. This fatty acid is that is saturated. What about stearic acid? That is also saturated. What about oleic acid? This is unsaturated fatty acid. And this is called monounsaturated fatty acid, mean only 1 double bond is present. So that's the 1 point to be noticed. Second, you can see that the palmitic acid, it contains 2 carbon less than oleic acid and the stearic acid. So that's why there's the 2 carbon are less in palmitic acid. So on the basis of this information, we're gonna see what is the correct option here. Now, statement number 1, the triglyceride POP contain 2 unsaturated fatty acid joined by ester bond to the glycerol. So this word 2 is wrong here. So you can see that in oleic acid, there is a 1 double bond is present. So that's why it's only 1 fatty acid chain, that is the oleic acid, which contain double bond. So this is unsaturated. So what about other 2 like palmitic acid, and we have the palmitic acid. So if there are the two palmitic acids are there and only one oleic acid, so this is only one unsaturated chain. So that's why this word 2 is wrong here. So option B, the triglyceride POS that contain 2 less carbon than to the atom the triglyceride POP. So for example, if you are replacing the stearic acid in this diagram with the P, so what will happen? So in this way, if we are putting 2P, so there will be overall, according to this structure, if this is present in POS, so if we are writing the POP, so what will happen? There will be this molecule, POS, will contain 2 more carbon, not 2 less. Why? Because the palmitic acid, this contain 2 less carbon in their fatty acid. So if you are using 2 extra chain of palmitic acid by the replacement of the stearic acid, so there will be not 2 less, there will be 2 more carbon in that. Okay, now the C option, the triglyceride SOS contain 4 more carbon atom than the triglyceride POP. So if you're replacing the P with the S, so if you're using 2 stearic acid, so obviously 2 stearic acid, if you're using, so each contain 2 carbon atom extra. So the 2 stearic acid as compared to the 2 palmitic acid, this will contain 4 carbon extra. So that's why C is our correct option here. Now the option D, the triglyceride SOS contain 2 saturated fatty acid joined by hydrolysis to the glycosidic bond. So in this way, you can see there are the 2 saturated fatty acids are there, S and S. So stearic acid are saturated, but that's joined by hydrolysis to the glycosidic, this is glycerol. So you can see here, there is one that's joined by the condensation reaction. So that joined by the reaction, this reaction is called our condensation reactions. So that is the difference. So here we need to change that. So this reaction is when the fatty acid is combining with the glycerol, so that is making a condensation reactions. So that's why this is hydrolysis. Word is wrong here. So we have a correct option. C is our correct option here. We're going to proceed to the question number 11. So which molecule contain at least 4 double bond? So we can see here, first we have an unsaturated triglyceride. This unsaturated triglyceride, so there are 3 carboxyl group they are present. So we studied like 3 fatty acid, they're combining with a glycerol and making 3 ester linkage. So there are 3 carboxyl group are present, and if it is unsaturated, so obviously at least 1 fatty acid chain contain double bond. So that's why at least 4 double bond are present in that. What about collagen protein? This is fibrous protein. And hemoglobin, this is globular protein. And these proteins are made of the thousands of amino acids. They're linked together in the form of monomer, and they're producing polymers. So that's why every amino acid which contain a carboxyl group, you can see here. So these carboxyl group, there are hundreds and thousands of the carboxyl group, they are present in that. So that's why you can see at least 4 double bond are present. So the D is our correct option. Why? Because all unsaturated triglyceride and hemoglobin protein and collagen protein, they all contain double bond. The question number 12, the diagram shows the amino acid glutamic acid. So there are 20 different amino acids that are naturally existing, and they have a different one thing, that is their alkyl group. What about the rest of other 3 groups are common? So for example, the central carbon in amino acid is called chiral carbon. This chiral carbon is connected with the hydrogen. There's one is hydrogen, second is carboxyl group, and the third is amino group. What about the one group which is different in a different amino acid? That is alkyl group. So here we have alkyl group. So what is the R group for the glutamic acid? So the R group is D, and this is our propanonic acid. So some R group are positive, some R group are negative, some are neutral. So that's why some protein that contain a different charge. So on the basis of these different charge, that's making the active side of different enzyme and giving a different property of different enzymes. So the D is our correct option here. The question number 13, the diagrams show the three interactions that hold the protein molecule in a shape. So we can see this is the tertiary structure of the protein. When the different polypeptide, two different polypeptides join together, that's making quaternary structure. So here we have interactions number 1. So in this way, amino group is having van der Waals interactions with a carboxyl group. Why? Because hydrogen contain partially positive, oxygen contain partially negative, and there is a force of attraction between these two polar molecules. So that is making hydrogen bond. And it's producing secondary structure in protein. Interaction number 2, you can see the two sulfur to disulfide interaction is that. So this disulfide bond is covalent bond. So there is a cysteine amino acid, which is making the disulfide interaction. So this is a covalent bond. What about the interaction number 3? Amino group with a positive charge and carboxyl group with a negative charge, and these are completely charged ions are there. These are molecular ions, and they're making interactions, and this is ionic bond. So these are 3 different interactions are there. So we're going to see that which option is correct according to. So interaction number 1. So interaction number 1 is our hydrogen bond. So that's our correct option. This is our hydrogen bond. Interaction number 2 is can be disulfide linkage, can be covalent bond. So that's why the option A and B both can be correct. So in the option number 3, you can see this is not a hydrophobic interaction. This is ionic bond. So that's why B is our correct option here. The question number 14. Some animals produce antimicrobial protein, which protect them from the pathogen. So these protein could be used to kill the human pathogen. However, when we use as a medicine, they are broken down by the protein digesting enzyme. So replacing one of the amino acid found in the protein with an amino acid that had been synthesized in a laboratory result in a modified protein that will be not broken down. So what could explain why this modified protein was not broken down by the protein digesting enzyme? So whenever this type of long questions come, you need to just overall go through this question, but move to the question statement and just thoroughly understand this question statement and mark that. Simply in this long statement, the examiner asking if you are changing the substrate, so what will be effect on the enzyme? So every enzyme has an active side, which is specific to their substrate, and they are complementary in a shape. But if you will change the enzyme active side, or if you will change the substrate structure, so they cannot combine together and they will no longer complementary. They will not make enzyme substrate complex, and they will not convert into the product. So that's simple as that. So we're going to see that according to this given information, which option is correct. So statement number 1, the modified protein has a different tertiary structure to the original protein. So that's saying like the new modified protein has a different structure. So it is no longer complementary to the enzyme active side. So that's why the statement number 2 is also correct. Statement number 3, the modified protein is unable to induce fit with the protein digesting enzyme. So it cannot fit into the active side of a protein digesting enzyme. So that's why these 3 options are correct. They are coherent of each other, and they are representing same concept, like when the active side of enzyme become denatured, when the substrate become denatured, then they are no longer remain complementary to each other. So they cannot make enzyme substrate complex. So if they cannot make enzyme substrate complex, no catalysis reaction will take place. So A option is correct here. The question number 15, a student investigated the effect of substrate concentration on the rate of enzyme catalyzed reaction. The graph was plotted to show that the relationship between these two variables, and the student was asked to take a reading from the graph that could be used to determine a Michaelis-Menten constant Chem value. So first we need to understand here what is Michaelis-Menten constant and how we can measure it. So first is a Chem value is a substrate concentration at which the enzyme work half of its maximum rate. So you can see that the enzyme is working at maximum rate when it is present on its middle way. So how we can find it? So the first we will find Vmax. So Vmax is a what? This is the value here. For example, if you see that the last diagram, so where the rate of reaction become horizontal. So this is called the saturation point. And this saturation point is Vmax. Now, that rate of reaction at the saturation point is a Vmax. So you need to identify where is a Vmax. That's the step number 1. And the step number 2, and then you need to find out half of Vmax. You can see in this next diagram in the left side. So this is the Vmax is above here. So this is half of Vmax is halfway of this. And the step number 3, we need to find out the concentration of the substrate along the x-axis. So where which is needed to get the maximum rate of enzyme reactions. So in this way, the question is, which 2 reading must the student use to determine the value of Chem? So if you want to find the value of the Chem, so step number 1 is a general word. What about the step number 2 and step number 3 is must. You have to take this step number 2 and step number 3. So we're going to see that where is the step number 1? For example, the step number 1 is here, the J. That's a general. Whenever the rate of reaction become horizontal, it means all enzymes get saturated, mean their active site is filled with substrate. So here the enzyme is no longer limiting, and that's going to be a substrate is enough for the enzyme. So and substrate are no longer the limiting reactant. So in this way, this is J is the point where is a V maximum. Now that's a general point. After getting the V maximum, you need to go for halfway L. So this L is must for you. Half of this V maximum is what? That's the point L. Now this L point, you will plot a line where it is intersecting to the graph line value. So then this line, you will drop it down. And at the point K, in the third diagram, you will find this value. For example, at the point K. So this point K, that is the substrate concentration at which the enzyme is working best. So that's a halfway. Michaelis-Menten says that the halfway of the V maximum value, for example, that one is a V maximum value, halfway of V maximum value, the enzyme is working best. So whatever the substrate concentration, which is required to get the highest speed of the enzyme, that is the Km value. So in this way, 1 point is L, second is K. These 2 are must for us to identify. So correct option for this question is here, that is C. So here we have done, we're going to proceed to the question number 16. The end product of metabolic pathway can act as a competitive inhibitor. This is called end product inhibitions, and this allows the cell to control metabolic pathway. So for example, if the cell is performing metabolic product, and how we can control the cell. So that's one mechanism which is present in cytoplasm of cell. This is called end product inhibitions. So if this end product will work as an inhibitor of enzyme number 1. So that's why all this series of action of enzyme will stop. For example, if the enzyme number 1 is here in the W, now product of enzyme W will be the substrate for enzyme X. And the product of the enzyme X is a substrate for enzyme Y. And in this way, this series will continue. Now we are going to have one statement here. And according to this statement, what would be effect if the enzyme Z was inhibited by the end product instead of the enzyme W. So for example, instead of using this end product in the enzyme Z, we can use this end product in an enzyme W. For example, if we apply this end product is here. So what will be impact? The impact will happen. Why? Because these intermediate enzyme, like enzyme number 1 intermediate, it will be working. So intermediate product, this is number 1, number 2, number 3, it will increase. Why? Because W, X, and Y will be working. So if instead of that, if you are applying here, so that's an inhibitor. Once again, I'm going to read the statement. What would be the effect if the enzyme Z was inhibited? So instead of inhibiting the W, we are inhibiting the enzyme number Z. So in this way, what will happen? So in this way, enzyme W is working, X is working, Z, Y is working. So all this and the intermediate product number 1, 2, 3 will increase. Why? Because these 3 enzymes are working. What about the Z is not working and end product is being used in the metabolism of your cell? So end product will be decreasing. What about there is no use of this is intermediate product of 1, 2, and 3. Why? Because these are not final product. So they will accumulate in the cell. So their concentration will increase, the intermediate. What about the end product concentration will decrease? So here, the option number A is correct here. The quantity of the intermediate product A, that will increase. What about the quantity of end product will decrease? So we're going to move to the question number 17 here. Which row show the distribution of cholesterol, carbohydrate chain of the glycolipid and glycoprotein in the cell surface membrane? So which molecule? We have one is cholesterol, one is when a carbohydrate linked with a lipid, that's making a glycolipid. When a carbohydrate bond with the protein, that is making glycoprotein. So which molecule is present on the cell surface? So cholesterol has a function to stabilize the fluidity of the membrane at high temperature and low temperature. So that's why it is present in the middle of the phospholipids. So you can see it is present in between of that phospholipid. It's not present on surface, mean near the head of phospholipids. So that's why option A and B are wrong here. The glycoprotein, when the carbohydrate is linking with the lipid surface or lipid head, so that's present on the surface, and mostly it's present on exterior or outer surface. Why? Because it is making cell-to-cell recognition point. So, and it's making cell binding site to another molecule, mean it's making receptor, it's making antigen, and that's why these receptor and carrier and antigen, they're present on the surface of the membrane. So that will present on outer surface. Number 1, mostly, and glycoprotein. So these glycoprotein act as a cell markers. So cell markers mean, for example, your white blood cell, they're identifying like which cell belongs to your body, which cell does not belongs to your body. So if any external tissue come inside of our body, so we get a tissue rejections. Why? Because glycoprotein is not mating to our body. So that's why this is a major problem, how we cannot donate we can take the body parts of donor to in our body. Why? Because there is a tissue rejection. The glycoprotein is not matching. So this is called cell marking, and that is present on mainly external surface of the membrane. So D is our correct option here. So cholesterol, this is present inside of the membrane, and glycoprotein, this is present mainly on the outer surface, and carbohydrate with the protein, glycoprotein, this is mainly present on outer surface. So these are the correct option. We're going to proceed to the question number 18. So which row correctly describe all possible relative concentration of the substrate when the substance is moved by endocytosis and exocytosis? So this endocytosis and exocytosis are a bulk transport. So this bulk transport, the endocytosis mean if the substance in a form of the bulk is moving from outside to the inside of the cell. So that is the bulk transport. And example is a phagocytosis, a white blood cell that is engulfing bacteria. And exocytosis, the waste material of the cell and enzyme secretion in the form of enzyme, they're releasing out by the exocytosis. So it depends only energy or ATP molecule. It does not depend on the concentration difference or gradient difference. So that's why endocytosis concentration equal, it's supposed to be. So what it does not depend. So it's the concentration equal or concentration inside and greater inside or greater outside. It will not depend on that. So the question statement is saying which row correctly describe all possible relative concentration. For example, if there's a concentration equal, this will happen. If the concentration greater inside, this will happen. If the concentration greater outside, this will happen. Why? Because it does not depend on the concentration gradient difference. It depends upon energy of the molecule, ATP energy. So if there's ATP energy is available, so active transport will take in place. So we're going to proceed to the question number 19. Which statement about the simple diffusion is correct? So A, it requires a specific molecule. So a specific molecule mean that is, for example, if you talk about osmosis, so a specific only water molecule moves through the partially permeable membrane. So that's for osmosis or chemiosmosis. For a chemical, they move to the partially permeable membrane down their concentration of the gradient. What about in a simple diffusion? There's no specific molecule. Any molecule can move by the random movement from a higher concentrations to the lower concentration in any directions. For example, the smoke particles are moving or Brownian movement of the particle. So that is the simple diffusion. So a specific molecule is wrong here. The B, it is a passive mode of transporting a substance. So this is correct. There's no need of energy in that. So B is our correct option here. It always requires a membrane. So there's no membrane is needed. The osmosis and active transport always require membrane. What about for simple diffusion? No need for membrane. It only happen the cell of the prokaryotes and eukaryotes. So this is diffusion and active transport. They, especially the active transport, it need the living cell. Why? Because it depends on energy. What about the simple diffusion? It don't need energy. So that's why the B is our correct option here. The question number 20. So the plant cell with the same water potential in their cytoplasm were each put in one of the three different concentration of the sugar solution. One is 10%, others are 5%, others 2.5%. So the cell were left for 50 minutes and then they were observed using the light microscope. So you can see that the first cell X, it has a normal shape. You can see it look like a normal shape. And this normal shape, this cell is present in isotonic environment. Outside, the concentration of the salt is outside is equal to the concentration of the salt inside. So that's why the net movement of water molecule moving from outside to the inside and inside to the outside is equal. In the same way, we have a Y cell. So it's a look like turgid. And this turgid cell is present in dilute solution, or you can say hypotonic solution. So hypotonic solution means inside is a more salt as compared to the outside. So the water will always move behind the salt. So this will move from outside to the inside and cell will become turgid. What about the Z cell? This is present in concentrated solution. We can say hypertonic environment. And in this way, the water move from inside to the outside and cell will get turgid. So we can say highest concentration of the salt is present in cell Z. Why? Because it has inside the high water potential. So water move from inside to the outside. So you can say the water potential level is inside is more, so it is less negative. What about the water potential is in the Y cell is a more negative inside. So that's why the water move from outside to the inside. And in the same way, that's in cell X is equal. So we're going to see that according to this given information, which option is correct here. So statement number 1, cell Y had lower water potential than the sugar solution it was put into. So cell Y has a lower water potential. Yes. So that's why the water moved from outside to the inside. So it is a correct statement. Cell Z was put into a 10% sugar solution. So it is correct. That's why it got plasmalized. And cell Z has less negative water potential. So then to the sugar solution, so it was put into. So less negative water potential mean that is low, that's a high water potential. So highest water potential is at 0. So if any, and the value of the water potential near to the 0, that is a less negative. So this less negative mean high water potential, and more negative mean low water potential. So that's why you need to keep in mind this terminology. So all these three statements are correct here. So A is our correct option for this. So which row describes some of the property of a stem cell? So stem cells have the ability to continuously divide differentiate into a specialized cell. So these the stem cell, they have a capability to divide. Why? Because they have their telomerase enzyme is continuously producing in these cell, and after division, when the telomere surface is decreasing, so they are continuously repairing their telomere. So that's why these cells can divide again and again, number 1. And number 2, after division, they are able to convert into a specialized cell. So which row is correct about that on the basis of this information? The ability to divide by mitosis and produce stem cell, it is correct. Second, able to differentiate into a specialized cell. This is a second characteristic of stem cell, it is correct. And able to repair the damaged cell. That is not a function of stem cell. So stem cell are not repairing the damaged cell. The stem cell are replacing the dead cell in your body. For example, you're replacing your skin continuously, you're replacing your blood cell continuously. So that means the stem cell are present at the base of your skin, stem cell are working in your bone marrow, and they're replacing your blood. So, but they are not repairing your damaged body part. For example, some people, due to the body trauma or some kind of accident, they lost their sciatic nerve and they got paralyzed, the lower body extremities. So if you lose your one time the nerves in your body, or by the polio infection, or by the accident of a lower in the extremities of your body, so you cannot repair it. So that is a permanent damage. So why? Because the stem cell cannot repair. So B is our correct option here. We're going to proceed to the next question here. The question number 22, the mitotic index is a measure of a proportion of a cell that are undergoing in a mitosis in an area of a tissue. It is calculated by using the formula that's shown there. The mitotic index is equal to number of cell undergoing mitosis divided by the total number of cell and multiply into the 100. That's a given a formula. Now, the scientists calculated the index of the area of an onion root at the different distance from the tip of the root. So we can see that highest number of a meristematic cell, which have a high dividing capability, and these meristematic cell are as like of stem cell in animal. So they have a high potency of the division and they differentiate to the other part of the cell. So they are highest in number, they're present in the tip region. If you're moving away from the tip region, their number of meristematic cell are decreasing, number of the cell division is decreasing. So we can have a proof on the basis of this graph. So you can see that if along y-axis we have a mitotic index, this is our dependent variable. What about along x-axis? And this is our independent variable. So that is our distance from tip of the root. So here, if you're moving from the distance away from the tip of root, number of cell divisions are decreasing. So overall, and the mitotic index is decreasing. So that's a general information. We got that. On the basis of this general information, we're going to see that which option is correct. So which statement is correct here? So A, no cell division occurs further than 1.4 millimeter from the tip of the root. So if you're going to see that from the tip of the root to the more than 1.4 millimeter, this became horizontal, and 1.6, 1.8. So that has a value of what? Mitotic indexes 2. So it means the still 2% cell are dividing after 1.4. So that's why this is mitotic index, it will be 2 here. So that's why it is wrong. The word no cell division, that is the wrong statement. The B, the rate of the cell division decreases as the distance from the root tip decreases. No, it is increases. For example, you can see that from the 0 to the 2. So if you are proceeding like distance from the root tip is increasing, the number of the cell division or mitotic index is decreasing. So that's why this word decrease is wrong here. Here's supposed to be increase. So C option, the most of the cell undergoing cell division are closer to the tip of the root. So you can see highest rate of index is where? So highest rate of mitotic index is near the root tip. That is the 12. So that's why this option is correct here. The D option for a sample 200 cell. Let's suppose we have a sample of 200 cell and we are in the position of 0.2 millimeter from the tip of the root. So if you're going to say that 0.2 millimeter, this is that's going to be on the tip of the root. So at the 0.2 millimeter, we're going to see that that's a value is going to be 2. So from the root tip, so we can find that, for example, let's suppose I'm going to put this value, you can see some calculation here, x divided by x number of cell we want to identify, divided by 200, multiplying to the 100 to get a percentage. So we will get our answer. That's 100 divided by 200. This will become x divided by 2. 2 is dividing, other side it will multiply, and we'll get a 2. So it means that at least there's still 2 cells are present. Or without calculation, directly you can get idea about that. At the point 2, you can see that, and this is the 2 cells are there. So what about 6 would be undergoing a mitosis? So the word figure 6 is wrong here. So C is our correct option here. The question number 23, which molecule make up the structure of ATP? So that is the adenosine triphosphate. And in this adenosine triphosphate, we can see the ribose sugar. That is a pentose sugar. And at the carbon number 2, you can see the hydroxyl group is present. So that is our ribose sugar. What about adenine base? There are 4 nitrogenous bases, and from these, one is adenine base because a minor group is present in the carbon number 1 here. So that shows this is double ring structure, and that is purine base, and this is purine, the sub is adenine base. Further, we have a 3-phosphate group are there. There are that's a triphosphate. So the question is, which molecule make this ADP? So adenine, number 1 is correct option, 4 is phosphate group, and 5 is ribose sugar. So in this way, 1, 4, 5, and C is the correct option here. We're going to proceed to the question number 24. A short piece of DNA, 19 base pair long, was analyzed to find the number of nucleotide bases in each of the polynucleotide strained. Some of the results are given there. So here, to solve this question, you need to understand some concept regarding the DNA. In the DNA, it consists of 2 strains, strain 1 and strain 2, and there are complementary base pairing are present. For example, adenine is present opposite to the thymine with a double hydrogen bond, and guanine is present opposite to the cytosine with a triple hydrogen bond. So in this way, how many number of adenine are there? The same number of thymine will be there in an overall DNA molecule. And the number of the guanine will be same to the number of cytosine. So on the basis of this concept, we're going to solve that. The first, we will complete this table. And this table, you can see some values are given, some values are missing. So we're going to complete this first value. For example, you can see in the strain number 1, the thymine are 4. So it means opposite to the thymine is what? Adenine. So statement number 2 will be 4. So I am writing here 4 with a blue color. Second, in the statement number in the thymine in the strain number 5, the strain thymine is 5. So adenine will be 5 in opposite strain 1. So I'm writing here with the blue color. In the same way, the guanine, we have what? Cytosine are 7 in the statement in the strain 2. What about a guanine in the strain 1 is 7. So according to this concept, you can fill this in the table. What about, we cannot fill here the cytosine. Why? Because we don't have the value of the guanine in the strain 2 and value of the cytosine we cannot have in strain 1. So we need to identify this value. Now the question examiner asking how many cytosine were in the strain 1. Now you need to calculate that. The further we will focus on 19 pair basis. So here we have a total basis is equal to what? The 19 multiplied 2 because they are present in a pair. That is equal to 38. Here further, now every strain, this is there is adenine is present on the strain 1 and strain 2. So that's why we will calculate this the two values of these each nitrogenous base. For example, to adenine, to cytosine, to guanine, and to thymine. So here there's a two value of adenine at 5 plus 4, and cytosine is that's a C plus 7, and guanine one value is present 7 plus G, and there is thymine one value is present, and that will be 4 plus 2 values are present 4 plus 5, and is equal to 38, and this will become our equations. Now for solving this equations, so we can put that value here. So let me have a bit corrections. Okay, so after solving this, we will get this answer. For example, this will become 9 plus C plus 14 plus G plus 9 plus is equal to 38. And that is equal to what? 32 plus C and G is equal to 38. Now here 32 here is a plus, other side that will minus, and that value will become C plus G, means cytosine plus guanine, that is equal to 6. And we know that the guanine and the cytosine are equal in number, so we will divide it to the 6 by the 2 to get the number of the cytosine, and answer will be 3 here. So in this way, we got our idea. So B is our correct option. And for your understanding, I'm writing in a complete long step for our calculation, but you're not supposed to waste your time in your exam. You can directly calculate and get your value. For example, if you calculate the total number, they are written there in the table. So that will be equal to 32. So if you directly minus this 32 from this total given number, so the answer will come 6. So it means 6 is the remaining two bases. That is the cytosine and the guanine. So 6 divided by 2, answer is the 3, and that will be your direct answer. So for your understanding, I did a long calculation, but you're not supposed to waste this time in your exam. So I'm going to proceed to the question number 25. Which statement about the role of the DNA polymerase enzyme in the process of semi-conservative replication of the DNA is correct? So what is the semi-conservative replication? The double strain of the DNA will split into the single strain, and every strain provide a template strain, and the nucleotide they arrange to the complementary base pairing, and when the new DNA will form, so half belongs to the parent strain and half is the consist of newly synthesized DNA strain. So that's why this is called semi-conservative replication. So on the basis of this information, we're going to see this, which statement is correct here. So A, the DNA polymerase form the phosphodiester bond. So that's a wrong option here. Hydrogen bond. So DNA polymerase is forming a hydrogen bond. It's not making the hydrogen bond between complementary base pairing. It is making phosphodiester bond between the nucleotide opposite to the template strain. So that's why this is a wrong statement. DNA polymerase move along the lagging strain in a 3 to 5 direction. So here in this way, the direction is wrong mentioned here. Always in a lagging strain and in the leading strain, the DNA polymerase always move from 5' to 3' in directions. So that's why it is wrong. C, the DNA polymerase join a new basis to the leading strain only. So word only is wrong here. The DNA polymerase is working with a leading strand, is working with a lagging strand, and it's also moving from 5' to 3' in directions. So on the basis of this information, the D is our correct option here. We're going to proceed to the question number 26. The diagram shows the process of translation occurring at ribosomes. So this is happening in the cytoplasm of cell when the translation has taken place. So messenger RNA with a codon sequence coming from the DNA from the nucleus. What about the ribosomal RNA? It will bind with the messenger RNA, help to read the transfer RNA. So why? Because the ribosomal RNA expose the codon sequence on the surface of messenger RNA. So in this way, the transfer RNA will bind to the with the anticodon sequence to the codon sequence and make a hydrogen bond. So you're supposed to identify the anticodon sequence at the place of S here. So opposite to the C, we have a guanine, a cytosine opposite guanine, uracil opposite adenine, and adenine opposite uracil. There's a thymine is absent. Why? Because this is RNA. So C is our correct option here. Question number 27. Which row correctly explain how the structure of the phloem C-tube element and the xylem vessel element are related to their function or transport system? So we need to see that the structure of C-tube element and the structure of the xylem is related to their functions. So to understand that, I shall proceed to one diagram here, and we're going to explain this diagram. You can see this diagram. We have a source cell that's to the extreme right, and we have companion cell beside to the source cell. We have a sieve tube element, and beside that, the sieve tube element, we have xylem tissues. So xylem and phloem tissue are moving side by side. So here, the source cell is producing sucrose, and this sucrose, this is first the source cell is converting glucose to starch, and starch is converting into the sucrose. Then the companion cell is performing the loading of a sucrose. How? So first, the companion cell is utilizing its ATP, which is produced in companion cell, and pumping the hydrogen ion in the cell wall between the source cell and companion cell. So the hydrogen ion are moving into the cell wall. When the concentration of the hydrogen ion increase in the cell wall, it start moving from high concentrations to the low concentration, and it start moving toward the companion cell. And in this way, they will open protein channel. When they will open protein channel, so the sucrose will use same protein channel. This is called co-transport. So, and it will enter into the companion cell. And that's the active loading of sucrose. When the active loading of sucrose will take place in companion cell, so this sucrose will mix with the water and move from a high to the low concentration into the sieve tube element by passive transport. And this is called facilitated diffusion. So that's our facilitated diffusion. So why? Because sieve tube element is connected to the companion cell by the plasmatosphere connection. So that's why they have facilitated diffusion. So when the sucrose will come inside of the sieve tube element, so in this way, there's a water potential will decrease. And when there's a water potential will decrease, so water will come from xylem to the sieve tube element through pit area. So you can see that the xylem wall are not completely the waterproof. So you can see that we have a terminology like water cannot move through the lignin, but water can move through the lignified wall. Why? Because the lignified wall contains pit area. So that's why the water can move through the pit area into the sieve tube element by osmosis. So here we understood that the role of the sieve tube element and the role of the xylem, and on the basis of this information, we're going to solve some question here. We are going to proceed on this question, and we are going to relate the structure of the xylem and phloem to their function. For example, the option A, the C-tube element contain mitochondria to make ATP for active loading of a sucrose. So you studied that the sucrose transfer from the source cell to the companion cell by active transport. But from companion cell to phloem sieve tube element, this is by facilitated diffusion. And this facilitated diffusion is not using. So that's why this ATP, which is being produced in the sieve tube element, is not directly used for active transport. So that's why that's option might be wrong in this way. Second is what? The xylem vessel that has a structure, what? The lignified cell wall. Now, the primary function of the lignified cell wall is what? It is providing mechanical support. So this mechanical support, that's a primary function. Now, the secondary function, it has adhesive property with the water as well. But the primary function is what? The lignin in the cell wall give a strength to the xylem vessel that do not collapse due to the transpiration pull. So that is mean this structure is related to this primary function. So that's why its function is wrong here. We're going to see that the B option, that contain no nucleus so that the dissolved solute can easily pass through. This is correct option. What about the xylem? The end wall between the two cells is perforated. The word perforated is telling like this contain the sieve plate, and this sieve plate is present in phloem sieve. What about the end wall is completely disappear? It's completely removed. So that's why this word perforated is wrong here. The C option, the end wall between the cell perforated. You can see this word perforated is coming in the phloem. This is correct statement. So that it dissolved, the solute can move between the cell. It is correct statement. What about the lignin, which is present in the xylem wall? That provides strength to the xylem wall and prevent from the collapsing. So that's why this structure resemble to its function. Second, the D option, no end wall between the cell. There is end wall present in phloem sieve tube element, and this is called sieve plate. What about the xylem vessel that contain no cytoplasm? So continuous column of water move, it is correct. So what about here? D option is wrong due to the phloem here. So C is our correct option here. So whenever these long questions come, you should take the pencil and try to identify or falsify these points on the basis of your the concrete concepts. We're going to proceed to the question number 28. Which component of the plant is used by apoplastic pathway as the water moves from soil to the xylem? So apoplastic pathway is what? When the water is moving from cell wall to cell wall. So Casparian strap. So these Casparian strap mean when the water will reach to the endodermis, and so there are Casparian strips, and these Casparian strips contain suberin wax. So that's why the water cannot move from cell wall to cell wall, and it has to enter into the cytoplasm of the cell as the symplastic pathway. So that's why a Casparian strap is wrong, endodermis is wrong, and suberin wax is wrong. Why? Because water cannot continuously move in the presence of Casparian strap, endodermis, or suberin wax. This is same place. So the same place has a three different component name. What about the water moving from cell wall to the cell wall? The B is our correct option. That is apoplastic pathway. Which statement correctly describe the transport pathway in a dicotyledonous plant? So we are going to describe what is the type of a pathway, which a statement is correct regarding to the given the pathway. For example, in a symplastic pathway, water may move through intercellular spaces. Intercellular spaces mean that the spaces between the cell. And the spaces between the cell is what? That is cellulose. So that's why it is wrong option. Here supposed to be a correct word was intracellular spaces, mean the water is moving into the cell. So that's why this is the wrong option. The statement number 3 is the symplastic pathway may be blocked by tonoplast. Now, tonoplast is a membrane which is present on the vacuole of the cell. That's present in the vacuole, that's present of the cell vacuole membrane. So that is tonoplast. So here, the symplastic pathway depend on plasmodesmata, the cytoplasmic connection between the two cells, or it is depend on cytoplasm. And it does not depend on the vacuole. So that's why there's no role of tonoplast in a symplastic pathway. And number 3, in an apoplastic pathway, the water does not move through the plasmodesmata. So water is not entering into the cell. So that's why this is our correct option. And the apoplastic pathway may be blocked by Casparian strap. Yes, it is blocked by Casparian strap. Why? Because it contains suberin wax, and this is present in endodermis. So that's why this is a correct statement. So D is our correct statement here. We're going to proceed to the question number 30 here. The plant such as the tobacco plant retain very little of water they take in, and the volume of water lost during the transpiration is very high. So that's why the transpiration is considered as that's the certain process in which you are getting advantage of taking carbon dioxide, but you have to lose the water molecule. So this is the kind of the drawback, you can say. And this is because this plant can only absorb carbon dioxide through opening stomata. So the plant can use the carbon dioxide to synthesize the glucose molecule, and it's estimated like 400 molecules of water are lost for each carbon dioxide molecule to gain. Now, how many water molecules are lost to produce a 1 glucose molecule? So we to solve this question, we need to know that this following information. For example, 1 glucose molecule consists of C6H12O6. This contains a 6 carbon atom. So each carbon dioxide contains 1 carbon atom. So to make 6 carbon atom, we need 6 carbon dioxide. So that's why to gain 1 carbon dioxide, we lose 400 water molecule. To gain 6 carbon dioxide molecule, so 6 multiply 400, that is equal to 2,400 water molecule will be lost. So that's why the D is correct option here. We're going to proceed to the question number 31. Which changes occur as the source is transferred from the leaf into the phloem sieve tube to transport it to the sink? So for example, if the source is transporting the sucrose from the source to the sink area, so what type of changes are taken place? So you can see that, for example, if there's the source cell is present, and this will transfer the sucrose to the companion cell. When this sucrose will transfer to the companion cell, so from companion cell to the sieve tube element, that is moved by facilitated diffusion through plasmodesmata. So when the sucrose is loaded into the sieve tube element, so the water potential of the sieve tube element will decrease. Why? Because high salt, low water. So in this way, the water will move from xylem to sieve tube element by osmosis. So when the water will move from xylem to the sieve tube element, so volume of the water will increase here. So here there's the volume is increasing. I'm trying to draw. So that's the volume is increasing here. So in this way, we can see, so when the loading will take place, so water potential will decrease, mean water potential will become more negative, and the volume will increase. Why? Because the water will come from xylem to the sieve tube element. And in this way, this will generate high hydrostatic pressure, and water start moving from a high to the low concentration. And in this way, this is called mass flow method. And in this way, we will get another concept. Now, if the distance from the source cell is increasing, the pressure, the overall pressure of water is decreasing. So that we will get in another question. Here, we are going to study the concept, which changes occur as the sucrose is transferred from a source to the sink. So the water potential in the phloem sieve tube element, you can see this will become more negative when the sucrose will be loaded in the sieve tube element. And then the water volume, it will increase. So D is our correct option here. Now, this is the next question. The graph shown does not have any axis label. So here there are two axes are there, and these axes are not labeled. According to the question, which rows show the appropriate label of axis that will be explain the mass flow in the phloem? So it's supposed to be what? One value is a hydrostatic pressure and other value is a distance from the sieve tube element. So we're going to get this idea. For example, if there's a distance from sieve tube element is increasing. For example, if this distance is present at a certain point, for example, if this distance is present at this location, so it will have a high hydrostatic pressure. So if this is present in this location, so this location has a high hydrostatic pressure as compared to this location. Why? Because the water is moving from a high to the low concentration. And in this way, the pressure is decreasing. So in this way, the location number 1, that will have a high hydrostatic pressure. And if there's a distance is increasing, so the hydrostatic pressure is decreasing. So it means the hydrostatic pressure depend on distance. So the distance is independent value, and hydrostatic pressure or water pressure in kilopascal is dependent value. So that's why we should know that value. So for example, here is going to be two values are given there. So we're going to solve this question. So along x-axis, there's always we put independent value. What about along y-axis, we put dependent value. So along x-axis, there is going to be a dependent value. So we're going to put that the independent value is the distance. So we are going to put along x-axis. What about along y-axis? That's our dependent value. So we are going to put this value along y-axis. So that's the dependent value along y-axis. Why? Because it depends on the distance. So as the distance is increasing in meter, so hydrostatic pressure in kilopascal is decreasing trend. You can see it's overall decreasing trend. So this way, we're gonna, on the basis of this information, we're gonna solve this question. So here we have along x-axis, there's a distance from the meter, and along y-axis, there's supposed to be a hydrostatic pressure. So C is our correct option here. We're going to proceed to the question number 33. What is present in the blood in human when? So that's a general question. For example, in the blood of human being, there is a chloride ion are present. They dissolve in a plasma, and to balance the ion in chloride shift in a red blood cell. Why? Because in the vein, the red blood cell is making carbonic acid, and then it will dissociate into hydrogen carbonate ion. And these hydrogen carbonate ion, when it will move out, so the chlorine will move into the red blood cell, and overall negative charge stay balanced into the blood cell. So that's present in the blood plasma. What about carbonic anhydrase? This is present inside of the red blood cell. And obviously red blood cell is present in the vein. So this is present. What about oxyhemoglobin? So the hemoglobin is present inside of the red blood cell, and it is carrying oxygen with that. So that is called oxyhemoglobin. So in this way, these three statements are correct here. So A is our correct option here. We're going to proceed to the question number 34. A photomicrograph shows two blood vessels, P and Q here. So you can see there's one blood vessel is the Q, so other is P. So the blood vessel Q contain thick wall. So due to the thick wall, we can predict this is artery. What about the blood vessel P? This contain thick wall. No, sorry, this contains thin wall. So you can see there's a less diameter. So that's a thin wall. So this is vein. So in this way, we're going to solve this question on the basis of this given information. So first, the artery transfer blood into arteriole, arteriole to capillaries, these capillaries to venule, venule to vein. So it means the correct option might possible is what? The artery transfer blood into arteriole, while the vein P get blood from venule. So that will be correct statement. So we're going to see that according to this information, what statement is going to be correct for us. So first, the blood vessel P, that is a vein. So the correct statement will be what? That carries blood from venule. So obviously it is the vein, it is carrying blood from venule. What about the Q is the artery? So this carries blood into the arterioles. So that's why this is C is our correct option here. We're going to proceed to the question number 35. The diagram shows the blood capillaries and the tissue fluid with which are surrounded in it. So we have the two regions. The first is arteriole end, and second is the venule end, and in the middle there's a blood capillary. So we need to identify the pressure overall present in the different locations. So I wrote it here with a red and a blue color. That's the pressure. So tissue fluid is a form when the fluid is coming from the capillary into the tissue. The blood when the plasma is leaking out. So that is forming tissue fluid. So we need to identify the pressure at the different location. But first, we are going to get some idea. For example, there's a highest pressure will be present at the point Z. Why? Because this directly, the blood is coming from ventricular systole. So highest pressure will be present at the point Z. And the lowest pressure will be present at the point Y. What about the pressure of W will be less than Z? And the pressure of the W in the tissue fluid will be same to the pressure of X. Why? Because the pressure in the tissue fluid is not changing. So that's why what is the value of the W? There will be value of X. So that's why we can get idea if the four values will be given to us in the exam. So highest value will be near arteriole. And the lowest value will be near venuole. And the same, there will be two value which will be same, that is of tissue fluid. Why? Because the pressure is not changing in the tissue fluid. So that's why here we have a highest value. So we're going to see that on the basis of the given option. Now, why there's a lowest value in the Y? Because all water will go out from the arteriole region to the tissue fluid. So only the larger protein like globulin and fibrinogen and hemoglobin and red blood cell, that will stay inside of the blood vessel. So that's why due to the high salt and there will be low water potential. So that's why there will be lowest pressure of water. So that's the reason behind, and we're going to solve this question. So you can see that and the pressure at W and pressure at X, that has a two value, 26 and 26. What about the pressure at Y? It's the lowest value, that is 18. So what about the pressure at Z? That is the highest value, that is a 36. So in this way, whatever the concentration are given there, so that's you cannot learn behind that all. So that's a concept is that the highest pressure will be near arteriole region, lowest pressure will be near venuole region. What about the tissue fluid? It has the two values that will have a same common number. So that's why the B is our correct option here. We're going to proceed to the question number 36 here. What happening during the ventricular systole in the mammalian heart? So ventricular systole means the ventricle is contracting and pumping the blood from ventricle to the aorta and pulmonary artery. So in this way, the aortic pressure is increasing. Why? Because the blood is being transferred from ventricle to aorta. So that's why if the blood volume is increasing and the pressure is increasing. So that's why the A is our correct option. Atrioventricular wall is supposed to be closed. Why? Because it's supposed to prevent the backflow of the blood. So if the ventricle is contracting, so atrioventricular wall is supposed to close. So here it's written open, it's a wrong option. What about the C, the semilunar wall, it's supposed to close. Because if it is, it's supposed to open. Why? Because if the ventricle is contracting, the blood is transferring from ventricle to aorta. So that's why this close is wrong here. Ventricular pressure is increasing. Why? Because if the ventricle is contracting, ventricle is in diastole. So that's why its pressure is increasing. So that's why A is our correct option here. The question number 37, the photograph shows a part of the human gases exchange system with one tissue labeled P. So we can see that it is a part of the human gases exchange system, which include the trachea and bronchus and bronchioles and alveolar duct and alveoli. So that's all is the trach here. Now in this trach, we can see that it is not trachea. And it is not bronchus. Why? Because there is a cartilaginous ring is not visible here. So if there is a no cartilaginous tissue, that's a one prominent cartilaginous tissue. So if it is not present here, so it cannot be trachea, it cannot be bronchus. So these are muscle at the point P, these are smooth muscles are visible. So that's why these are smooth muscles. So this is our bronchiole. Now, these smooth muscles are doing what? And they are controlling overall the amount of air is moving into our breathing trach by controlling the diameter of the bronchiole region. So on the basis of this information, we're going to solve this question. Which row is correct here? The part of the gas exchange system, so that is A and B, these are bronchus. That's wrong. Why? Because cartilaginous tissue is not present. So that's why A and B, these two parts are wrong. What about bronchiole is correct here? So C is our correct option. It regulates the amount of air reaching the lungs. Why? Because when there's a contraction of the muscle, it's decreasing the diameter of the bronchiole. So that's why it is regulating the amount of air is moving into the lungs. So that's why the C is our correct option. What about the bronchiole? And this smooth muscle provide a protection and it prevents from a collapsing. This is function of the cartilage in bronchus and in trachea. So that's why this is a wrong function of smooth muscles. So C is our correct option. We're going to proceed to the question number 38. How many times must a molecule of oxygen pass through the cell surface membrane as it diffuses from the air spaces into the alveoli through the cell in the capillary wall to bind to the molecule of hemoglobin? So when the cell is moving from the alveoli to the hemoglobin, so we're going to take it past. For example, that there are two cells are involved here. The one cell of alveolar cell, second is the capillary cell, and the third cell where it will enter, that's a red blood cell. So first it will enter in the alveolar cell and cross one cell membrane. Then it will exit the alveolar cell and it will cross second cell membrane. Then it will enter the capillary cell and this will cross third cell membrane. And then it will exit the capillary cell and its fourth cell membrane. And then it will enter into the red blood cell. This is fifth cell membrane. So that's why overall it will cross 5-cell membrane. So here the C is our correct option here. We're going to proceed to the question number, there's a 39. A student wrote 4 statements about cholera. Which statement is not correct? So here they're writing not in a board letter. Why? Because most of the students, they cannot perceive the note and they start moving in a wrong direction for answer. So always you're supposed to be careful in the question, where is it written not, where is it written always, where is it written only. So these are the indication for answers. So A, it can be controlled by the vaccine. So yes, there's a vaccine is available for cholera, and the vaccine is providing artificial active immunity by providing actual antigen. So second B, it is caused by bacteria that's spread by ingestions. So Vibrio cholera bacteria, which is contaminating the water, and it transferred by ingestion process, mean by drinking. HIV, that is human immunovirus, and that is acquired immune deficiency syndrome, increase the risk of infection. So whenever there's an AIDS disease, it's basically the immunosuppressant diseases are there. So whenever that AIDS virus is infecting the body, it is basically decreasing the white blood cell T4 cell, which is increasing immunity. So if the person immunity is less, so risk for this disease will be more. So that's why this is correct statement. The D, it may be transmitted by animal vector. So this animal vector is wrong. This is the disease is malaria, which is transferred by a mosquito as a vector. So that's why this is a wrong option. So cholera is transmitted by contaminated water. So that's why the D is our correct answer here. We're going to proceed to the question number 40 here. Monoclonal antibodies are produced by using hybridoma cell. To understand the concept of monoclonal antibodies, we are going to understand this concept here. For example, if you want to produce so many antibodies regarding one specific or particular antigen, so we take that antigen from pathogen or from bacteria or from virus, and we inject that antigen into the into the mouse. So this mouse immunity system start producing the antibodies against antigen. So that's why this mouse survives. But with that, it will produce the plasma cell, memory cell. So these lymphocyte cell or plasma cell, they will be present in spleen of the mouse. So if the mouse will survive with this antigen after a few days, so we will collect the lymphocyte cell from the spleen of this mouse. But the problem is that these lymphocytes, they are specialized cell. They cannot divide. So that's why we need to put that dividing capability in these cells by combining them with a cancerous cell. So when we combine these lymphocytes with cancerous cell, we are producing hybridoma cell. Now these hybridoma cell, they have a capability to divide, and they have a capability to produce monoclonal antibody. So that's why these cells will divide by mitosis and produce so many monoclonal antibodies. And these monoclonal antibodies, they have a same chemical nature and which are effective against that antigen which we entered in the mouse. So that's why this is all the process for creation or for production of monoclonal antibody. So here there's a question, what was removed from the spleen of mouse to produce monoclonal antibody? So here we remove from the mouse is what? That is lymphocyte. So we remove the lymphocyte from the spleen of the mouse. That is the correct option. So we have completed our this paper. So hopefully it will be helpful for you. If you didn't understand any questions, so you can write your doubt in the comment box. I'll try to answer your questions. So thank you. Thank you very much for attending the session.